Thank you very much for this opportunity to speak to you after such an illustrious group of speakers. I've got a tough, high bar to reach. I'm afraid I'm not going to talk about therapeutics because, as Stuart pointed out, much as a neurosurgeon would love it, you can't regenerate the brain. But what I'd like to show you is that cerebral organoids, which I've become involved in over the last 3 or 4 years, have a great purpose in understanding disease mechanisms and disease modeling. My starting point and my year zero is also 20 years ago, but with microcephaly, which I've worked on genetic forms of that for 20 years. I guess for most people, year zero is this year, with the Zika microcephaly making this a disorder which needs no introduction. So, my interest started from this. Brain size varies greatly over between mammalian species. And I guess I should point out our favorite model organism here. Let's find it, mouse, just hidden down here, compared with humans. And there's a 10,000-fold increase in cortical surface area between mouse and human. So there's a huge scaling in this organ that we think defines us as humans. I guess we should point out that dolphins and whales are also pretty good in the brain department as well. So this scaling has come about, we believe, predominantly through an increase in neural cell number, such that we all each have 100 billion neurons in our adult brains. How the cerebral cortex develops is as an outpouching at the rostral end of the neural tube. Topologically, it remains a tube, such that there's a fluid-filled center, the ventricles, and the grey matter is around the outside. In the developing brain, during neurogenesis, when all your neurons are generated, the neural stem cells or neural progenitors lie at this inside surface next to the ventricle. This is the apical side of this neuroepithelium. And then as neurons are generated by these stem cells, they migrate along a radioglial scaffold to the cortical plate, which then becomes the neurons, 6-layered neurons of the cerebral cortex in the adult brain. So during neurogenesis, there's a rapid expansion in cell numbers through symmetric cell division to generate daughter neural stem cells. Subsequently, there's a shift to asymmetric cell division where daughters of different fates are generated. And this one remaining a stem cell and one becoming a neuron, which then migrates. This shift is believed to result from a shift in cleavage plane from exactly perpendicular to the ventricular surface to one which is oblique. And the reason this is important is that there are basal fate determinants located at this region here, such that if you cleave like this, you end up with one cell retaining basal fate determinants and stemness, whilst the other then adopts a neural fate. So it's believed that there is that the dynamics and the shift between symmetric and asymmetric cell division may underlie the expansion, cerebral cortical expansion that's occurred, in that if you remain longer in this state, you expand your stem cell pool and then eventually generate a larger amount of neurons once you shift to the asymmetric state. So that's evolutionary. From a disease point of view, my doctoral work was in a condition called primary microcephaly, which in essence is a single gene disorder, autosomal recessive inheritance, which causes the human brain to be a third of normal size. So that's comparable to the brain of humans that existed 2 million years ago. It's a disorder in which apart from size and some intellectual disability, there are no other neurological deficits or abnormalities in other organ systems. Shown here is a sagittal MRI section, taken if you imagine a meat cleaver cleaving me in half. This is an adult female MRI scan of a normal individual, and the cerebral cortex is this region here, and you can see the multiple folds in here. And this is a lady with a mutation in the MCPH1 gene, the first gene that I identified 15 years ago. And as you can see, the dramatic change in size is predominantly a reduction in cerebral cortical size, along with a simplification of a gyral folding. So over the years, my lab and also many other labs have identified a large number of genes for microcephaly and also microcephalic dwarfism. In terms of microcephaly, I guess I should stress the contribution of Jeff Wood's lab to identify many of these components. The other remarkable thing about many of these components is that they encode centrosomal proteins. So the centrosome is a key cellular organelle which is important for mitosis, and its role is in nucleating the mitotic spindle, such as can be seen here. Consequently, it's believed that microcephaly, or it's been postulated that microcephaly is a neurogenic mitotic disorder. And the predominant model in the field has been that as centrosomes nucleate the mitotic spindle, they'll also determine its orientation. Consequently, if you have dysfunction in the centrosome, you will shift from being able to maintain a perfectly perpendicular cleavage plane to this being randomized. As a result, you will undergo premature shift from symmetric to asymmetric cell division and end up with premature neural production and exhaustion of your neural stem cells, reducing cell number and reducing brain size. So a lot of our work in the lab and functional work has been done on cells which are available to us, and in studying humans up until recent years, we've been restricted to blood cells and skin cells, so fibroblasts. And much of my work to date has been on such cell types, and we've been fortunate that most of the genes we work on, or most of the microcephaly genes, are ubiquitously expressed in all proliferating cells. However, there are certain questions, such as the shift in cleavage plane, which can't be answered in a 2D cell culture system. Similarly, available developmental models are less than ideal for this. For instance, zebrafish, their forebrain is a sixth of the size of their eyeball. A mouse has a smooth cerebral cortex, which I've also said is dramatically reduced in scale compared with that of a human. And this may explain why as many of the mouse models of microcephaly have had very subtle or non-evident phenotypes when their genes have been knocked out. So the advent of reprogramming technologies and iPS cells and the ability to derive neural stem cells had caught my attention, but it really wasn't until Jürgen Noblik and Madeleine Lancaster approached me with the system that they had developed that we really got engaged in trying to model microcephaly in a relevant tissue setting. So Madeleine had made substantial advances on the work of other groups, including the SASI lab, where the differentiation of pluripotent stem cells, either iPS or embryonic stem cells, into embryoid bodies and down a neural lineage, had been extended to take these cells and embed them within matrigel droplets and then place them in a spinning bioreactor. And using this, she was able to generate small tissue aggregates, three-dimensional aggregates, which are up to a few millimeters in diameters. And these don't look terribly remarkable externally, but when they're sectioned, you see a very striking phenotype of a complex tissue structure with many features which will be reminiscent to those of you who work on mouse or other vertebrate neurodevelopment. So, for instance, regions which resemble forebrain, presumptive cerebral cortex development, midbrain, hindbrain, or choroid plexus. So these structures also, with the term mini-brains, recapitulate many of the features of early human development. I think it's important to emphasize at this point that this is not a perfect recapitulation, particularly in terms of organization. Here is an example of an embryo, a human embryo at a similar stage, where you can see that development manages to do it in an accurate temporospatial pattern, which isn't perfectly recapitulated on a recurrent basis in these organoids. However, the important thing, and particularly for microcephaly, is that in almost all organoids we see areas which have presumptive cerebral cortex, which we can study the cell biology of neural stem cells. And in this context, these organoids are amazing in their ability to recapitulate early cortical development, the inside-out arrangement that you see during mammalian neurogenesis, with stem cells marked in red on the inside and neurons at the out, is recapitulated in the organoids. Not only that, but many of the cell behaviors which are seen in early neurodevelopment are also present. So, for instance, mitosis occurs at the ventricular surface, as occurs developmentally. The process of nucleokinesis, which is the migration of a nucleus during different cell cycles, also occurs. The neurons have electrical activity. I won't show you that. And there are also human-specific features to these cerebral organoids, including the presence at reasonably high frequency of basal radial glia, which are thought to be a property of gyrified mammals. From our point of view, given our understanding of primary microcephaly as a disease of neurogenic mitosis affecting stem cells, we thought this would be a system which could be modeled, which we could use to model disease. And our starting point in our collaboration with Madeline and Jürgen was with this patient who has an absolutely extreme microcephaly, as you can see here, in terms of quantification. Head size was minus 14 standard deviations below the population mean for her age, so absolutely extreme. She had a null mutation in this centrosomal gene, CDK5-RAP2, which you can see in immunoblotting of fibroblasts, and also immunofluorescence led to complete absence of this protein. And these cells, fibroblast cells, were reprogrammed into iPS cells, and Madeline then differentiated these into organoids, and there were immediate differences apparent, including the premature differentiation at early time points. So this is day 22, with more differentiated neurons in green here, compared with progenitor cells, stem cells in red. Not only that, but she observed that there was a alteration in cleavage plane in these early neural progenitors, such that instead of the predominant perpendicular cleavage plane seen in control organoids, she was seeing essentially a randomization in cleavage plane occurring, suggesting that this cleavage plane and therefore asymmetric fate, assumption of an asymmetric fate and neural differentiation explained the phenotype she was seeing in the organoids of premature accumulation of neurons, which would eventually lead to a reduction in brain size. So just schematically to summarize this, instead of a large population of stem cells being generated, which then subsequently went to generate a large number of neurons, the premature generation of neurons led to an eventual depletion of neurons, explaining the reduction in brain size that we saw in our patient. So that work was published a couple of years ago now, and I thought I would just share with you what we've been doing in our own lab since to try and utilize this technology further and put it in the context of the other tools that we're doing, both basic cell biology using conventional 2D culture techniques, both in primary fibroblasts and considering other neural stem cell mechanisms, but also in terms of developmental organisms, including mice. So one of the genes which we've taken next to study is this gene PLK4, which we recently identified as a novel microcephaly gene causing extreme reduction in brain size, as you can see here. This gene is also the master regulator of centriole biogenesis, and centrioles stand at the center of the centrosomes, so they're a key centrosomal component. Another interesting facet of this work is that the major mutation we identified, which is a homozygous frameshift mutation, is partially rescued in primates and in apes because of an alternative splice form. And therefore, this mutation would be difficult to model in rodents. Therefore, we took fibroblasts from two independent patients from two different families with this mutation and reprogrammed them using conventional IPS reprogramming techniques and generated PLK4 organoids. We again observe a reduction in progenitor cell number as, and again, it's harder to see here because of the lighting, but essentially as measured through the thickness of the ventricular zone, of the thickness of numbers of progenitor cells, SOX2 positive cells. And this we expected to be the consequence of altered cleavage plane. And as you can see here, this wasn't what we've seen. Therefore, in this model, also due to a centrosomal gene mutation, in fact, it's not the cleavage plane that is explaining the premature depletion of neural stem cells, and this is something that we're actively additionally investigating now. The other thing I just wanted to touch on is that cerebral organoids are a fantastic addition to our armament of studying the neurobiology of microcephaly. Their limitation at the moment is that they model accurately early development. In the paper we co-published with Madeline and Jürgen, Madeline was able to generate early cortical plate, and subsequently other groups are now able to generate a 6-layered cortical plate. But these remain early, a model for early neurodevelopment, and therefore limit their applicability to more general, to other neurodevelopmental and adult onset disorders. The other issue is, as it's been alluded to, they're very labor intensive. You need a lot of patience and a lot of levels, particularly time. I mean, these are relatively early organoids at day 45, day 70. It's a long time for your experiment. The last key issue that we face is that there is a variability from organoid to organoid in the amount of forebrain tissue which is generated, which makes quantification challenging. There is issues with batch-to-batch variation. There are other variabilities which have been alluded to previously today, such as clone-to-clone variation in neural differentiation potential. So there are a lot of issues and challenges still to be faced. I think where they sit best is at this interface between cell biology and developmental biology. And for my thinking, this, for me, is the ideal system to be studying neural stem cells as a cell biology question in a near physiological environment, rather than a developmental one. And I think mouse disease models still have a place to play, and they can be informative, as our recent work has shown in other models. So in summary, I want to finish by saying that the reprogramming technology has now, and advances from that have now provided us with much more sophisticated cell culture models. These 3D models are physiologically much more relevant to early human development, and these are giving us the opportunity to study neural-specific features of primary microcephaly to gain greater insight. And I thank you for your attention.
So you said something there?
Yeah, no, could I just acknowledge a couple of people? Carol Ann, who's done all the organoid work in my lab, and also I should emphasize the huge contribution of Madeline and Jürgen to the work I've been presenting.
Great. So just while people are thinking of questions, could I ask if we were to dare to think about a therapy for people with poor neural development, brain development, do you think we could think about either stem cell transplantation, or do you think we could seek to control the proliferation and differentiation of neural stem cells in children that had an early diagnosis?
So the major issue is that neurogenesis is confined to a very small developmental window in early fetal life, so that by 16 weeks you've pretty much generated all the neurons you're going to generate, apart from a few small areas of the brain. So you'd have to intervene prenatally, and I think that's what limits the therapeutic opportunity here. I think being able to... So
you don't think that we'd ever think about transplants or doing something to promote a division of, to be trying to de-differentiate cells? That would be too...
I think, I mean, I don't know, I'd love to be surprised. I think particularly with the huge complexity of the brain following the generation of neurons and their post-differentiation and wiring, I find it hard to imagine and contemplate, but maybe in 20 years' time we'll think differently.
Question?
You mentioned Zika in your introductory remarks. Any evidence yet that the virus is directly interacting with components of the centrosome system?
So one of the things I meant to emphasize as I went along is that not all microcephaly is the same. So microcephaly literally means small head. It's never due to problems with the skull, it's due to the underlying brain. But what happens to the underlying brain is very different in Zika. I actually have a picture somewhere along here. So essentially in Zika, you've got a small head, but you've also got other processes going on. So there is a small brain, and there may well be a direct pathogenic effect on neural stem cells, reducing cell number. But very strikingly, there's a large amount of intracranial calcification which occurs in Zika virus patients, similar to that seen in cytomegalovirus and other congenital viral infections. And I think there is an innate immune response going on, not necessarily in neural stem cells, and hence the question mark in my original title. I don't think all microcephaly is going to be a stem cell disorder, but there may well be a component in Zika.
Okay, Sally, quick question, quick answer, please.
So it was about your mutant where you interfere with centrosome biogenesis without affecting the cleavage plane. So do you know what's happening with the cilia? Do you think you're interfering, for example, with hedgehog signalling, and that might explain the phenotype?
So we've also, work I didn't present is we've also modelled the PLK4 mutations in a zebrafish model, and we actually see there's a separation of separation of function is not quite the right word. Developmental separation in terms of reduction in PLK4 activity. So in our patients, sorry, this is not a short answer. You reduce PLK function to about 10% of normal. At that level, you get just an effect on mitosis, and I think the predominant thing which is happening there is you're getting a delay in mitosis, and that may well be influencing differentiation state. If you deplete further, then you start to get ciliopathy-like phenotypes. Because one of the things which has always intrigued me about these disorders is why the hell don't you get ciliopathy features? Because centrosomes form, and centrioles form the basal bodies for cilia as well. And I think that's the explanation, is a differential sensitivity.
I think in the interest of time, we'll have to curtail questions, and thank you very much for that.